All right, medical interventions for December 7th and December 8th. I'm going to talk through some new stuff that we're going to be doing. If you remember last week, we talked about the structure of, we did some research on bacterial structure, gram staining for gram-positive, gram-negative bacteria, and how and the different classes of antibiotics and which might be most helpful in treating the illness that Sue has and some of the others have with this meningitis bacteria. So this week, we're going to focus on antibiotics and how they function, and then also which antibiotics to choose in certain situations and how different ones react, as well as how antibiotics, if used too much, can cause resistance of the bacteria to where they don't function correctly, and the antibiotics are no longer effective or less effective. All right, so I'm going to do a little bit of instruction on here as well of using a drawing that I'm going to be doing, so you can add that to your lab notebook as well. So we're going to go over a lot of the 1-2-1 workbook in class. So we'll cover a lot of that in class on either Monday or Tuesday, whatever day you're there. I threw a lot of information at you guys last week, but we really haven't covered it. So we're going to cover some from 1-1-6 and 1-2-1 and make sure we're really clear on that before we move forward. So we're going to do that. Additionally, we'll be starting a new lab titled Which Antibiotic is the Best Choice. So I'm going to start there. Which antibiotic is the best choice? So there's different classes, and you understand that Neisseria meningitis has the gram-negative characteristic, and so you would use a class of antibiotics that would be more effective with that. But if you want to see if something, a new bacterium, is affected by antibiotics, then you can do a simple test called a Kirby-Bauer test that you're going to investigate. We're going to do this real simply in the class. We're not going to do a huge write-up on it. You'll be using your lab journals for the first time to write down some data on that in class. So when you're in class Monday, Tuesday, you'll be drawing out those the slides that we do in this activity. So you're basically going to be taking a slide with bacteria in it, but the bacteria have not really grown much yet. They're like within the broth, within the gel. And then you're going to put discs on it with different antibiotics in those discs. So each disc is a different antibiotic. One might be penicillin, one is ampicillin, streptomycin. There's different names for these. And those different antibiotics we're going to put onto the dish, and we're going to let them incubate. And so you'll do that Monday or Tuesday, and then Wednesday or Thursday you'll see the results, because then you have to allow the bacteria time to grow. And what we're going to try to do is observe, do those discs inhibit or prevent the growth of the bacteria in that area? All right, so that's going to be that part's going to be in class. There's not much to write up about that. There will be a drawing that you'll do in your notebook. Now, there is a student workbook assignment. This is only digital, so it's not going to be printed out. 1-2-2, which antibiotic is the best choice? So this is something you should be working on the day that you're not here. You will not work on this much in class, all right? And this is due Thursday to the 10th, so you do have quite a bit of time to do it. So which antibiotic is the best choice? So if you click here, and I made it due a little bit later because the lab takes a couple of days. So this is the 1-2-2 activity. Basically, you're going to read through here. You may need to zoom in a little bit on some of this. If you just hit like Control+, it will typically zoom you in more to where you can read that a little bit better. And it is copied from another text, so it may be a little bit more difficult to read. So you're going to read through this, read the paragraphs, and then you're going to paraphrase how a Kirby-Bauer test is done. All right, follow the directions there. And then you're going to look at a patient case. There's some background about someone named Nico, a boy named Nico, and he had a surgery for his appendix. And we get into antibiotics. I'm going to have you read through that, antibiotic resistance. So basically, antibiotics, if they're prescribed over and over again, eventually bacteria get mutations during their replication of their DNA that randomly they get a mutation that allows them to be able to survive or be less affected by the antibiotic. And so then they can, because of that, they can survive. And then that antibiotic is no longer effective in that situation. We call this antibiotic resistance. You hear about things like called antibiotic resistance. We hear about things like MRSA. It's methicillin resistant Staph aureus, Staphylococcus aureus. So it's a staph bacteria, Staphylococcus bacteria, but it's methicillin resistant. Methicillin is an antibiotic that normally can kill the Staphylococcus bacteria, but it is methicillin resistant. So it's harder to destroy. All right. So there's some questions here. You're going to read through after you read this information, the background, and then answer these questions. Just type your answers. Since these are like screenshots, you're just going to type your answers to the questions here, and then you're going to look at a sample of a lab activity and read through here. And for each one, you're going to answer the questions. And so you have question 1, and then questions 2A, B, and 2. There's no question. I think I messed this up here, so I'll change the document here. So yeah, they're. Let me just delete this row. Modifying on the go, and this is 2A, 2B, and 2C. All right, because there is no 3. And so I think I just numbered it wrong. So you'll read through the results here, and here you can see those discs and what's called the zone of inhibition around them. And so here, this where it's cloudy is all growth of bacteria. But here you can see in this area around the bacteria or around the disc of antibiotics, you don't see much growth. That would indicate that this is very, very effective. Whereas if there's no ring, that means it's not effective against the Staphylococcus pyogenes or Streptococcus pyogenes in this case. So it would not be effective if there's not a zone. This would be something that would be effective. Okay, so you answer those questions and then submit that. Again, that's due Thursday, but that's going to be something completely outside of class to do. All right, and so the only other thing is you're going to get in class on Tuesday or Monday. You're going to get a new packet called the Attack of the Superbugs packet. And so you're going to get this packet, and there is a diagram in here. You're going to make a list of 3 things that can be done to reduce antibiotic resistance. You don't need to do this yet. What I want you to do, though, is to get a little background. This is where I'm going to talk through this for a minute. So in your lab notebook, to get some background on this, I'm going to discuss this. You'll need to fill this part in, 3 things that could be done to reduce antibiotic resistance. You'll need to research that. So what we're going to look at is kind of organizing and getting the background about why it's important to know about antibiotic resistance. So I'm going to draw this out real quick and switch to this overhead camera. This is not my normal overhead camera. This is a document camera, so the image may be a little bit more grainy than you're used to, but I think you should be okay. Probably the worst part is going to be my handwriting. All right, so with this, make sure I'm still recording here. Yeah, okay. Okay, so we're going to start with a very common bacterium named Streptococcus pneumoniae. All right, so Streptococcus pneumoniae. So that's our first. So this is a very common bacteria. It causes strep throat, causes other respiratory infections. Okay, and it is usually treated with the most common antibiotic. You guessed it, penicillin. I'm sure I didn't know if you actually guessed it, but penicillin is something you probably heard of. Amoxicillin is maybe when you're younger. Younger children tend to get ear infections, so maybe you got ear infection as a kid. Penicillin is that like bubblegum flavored kind of pink medicine, has basically amoxicillin or penicillin in it. All right, so typically you're going to treat it with that. And once again, it is a bacteria that causes disease like, and if you look at the name of it, pneumonia. Okay, pneumonia, which is basically a bacterial infection of the lungs. And so now we have, so this is like what normally happens. You have this bacteria, it causes pneumonia. We usually treat it with penicillin. However, this has been around for a while. Penicillin's been around since the 1930s. Now this is developing, over time, is developing antibiotic resistance, meaning the bacteria is becoming resistant to the antibiotic. It's not being effective in killing it or preventing its growth. Some antibiotics work by killing, some work by preventing growth, right? So it, turn that light back off. All right, so, and when it does this for a long period of time, it is now turning into what we call a superbug. And you would think that'd be a great thing, but it's not. It's not super at all. These superbugs are pathogens that are difficult to kill. All right. So the superbug in this case is the Streptococcus pneumoniae bacteria is becoming a superbug because it's antibiotic resistant. All right. And so this is one of the major health issues in the world, or really in the United States, but really everywhere, according to the CDC, which is the Center for Disease Control. Center for Disease Control, which is a government organization that helps prevent the spread of disease. You've probably heard about them over the last year. All right. So, are there other superbugs like this, though? Yes, there are some other ones. So, superbugs are also associated with a couple of other illnesses. So, also associated with things you probably heard of, things that you, so like tuberculosis. We don't get, if there's a case of tuberculosis in the United States, it would be huge news. All right, people are vaccinated against tuberculosis. People just don't get it here, and because we get vaccinated, but the antibiotics that work on tuberculosis aren't as effective because a lot of the tuberculosis bacteria are resistant. Also, gonorrhea, which is a sexually transmitted disease, is another example of one that is the antibiotics for it are becoming resistant, or the bacteria becoming resistant to the antibiotics. And so, how does this happen? Um, so, how do you get one bacterium to make, like, so let's say one gets a mutation, um, in that allows them to be antibiotic resistant. Well, that bacteria can multiply and have more of its own offspring, which would then be antibiotic resistant, right? So normally you have one cell becomes two, right? That's normal, right? You have cell division. But sometimes cells can actually, one cell can give its DNA to another cell, all right, and cause that. So this resistant one can make this one resistant by passing its DNA to it, all right? And so the superbug can kind of spread. So it spreads between bacteria by the transfer of DNA. Okay? And so here's our DNA molecule. All right? Maybe right here is that gene. So this is the antibiotics. ABX is abbreviation for that. Antibiotic resistant gene. All right? So if one bacterium, if this bacterial cell is resistant. Okay, so let's say this one is resistant to so penicillin resistant. Okay, based on its genetic code, right? And it happens to get mixed in the same area with another one that is sensitive to it on its DNA. So this one has a special little gene on there. Then what can happen is we can have a transfer of that. And then the offspring of this one are resistant, and the offspring of this one are resistant. So now you have another strain of bacteria, another version of the bacteria. They're now becoming resistant just because these two happen to be close to each other. All right, so that resistance can spread lots of different ways. So that's sort of some background. And so if you'll draw that out in the lab journal, that'll give you some good background information, because we're going to go into another lab on Wednesday, Thursday, where you're going to work through, I'll put the picture of this in Canvas as well, so you can zoom in on it a little bit more if you need to. But we're going to do a superbugs lab where we're working with 2 strains of E. coli bacteria, and we're going to be working to see if we can convert one strain that is resistant or that is sensitive to it to become resistant to the antibiotic. So to sum up, close down my camera. All right, to sum that up, things that you need to turn in. Which antibiotic is the best choice? That's what you're working on outside of class. You should have everything else turned in up to this point, prior to this week. And then we'll be working on this lab and the Attack of the Superbugs lab in class this week. You'll have some research and some things to do later in the week. I'll probably throw in one more activity, which is the 1-2-4 activity that shows how giving too much of an antibiotic can cause problems over time. So that is all, and I will see you soon. Thanks.